ஹலோ. சொல்லுங்க. காமராஜ் ஹாஸ் நீங்க எங்கண்ணா இருக்கீங்க? காமராஜ் ஹாஸ்பிட்டல்ல வந்துருக்கேன். நீங்க எங்கண்ணா இருக்கீங்க? சரிண்ணா. சரி. எங்கண்ணா பேக்கேஜ் என்ன இது? பேக்கேஜ் யூஸ் பண்ணுறீங்க? ஓகே. கடைசியாக ட்ராப் எங்கண்ணா வரும்? சரி, ஓகே. சரிண்ணா, ஓகே.
எப்படி பண்ணு போயிட்டு ரைட்டு நானும் ஹோமியோபதி இங்கதான். எவ்வளவு ஆச்சு?
217 ரூபா மேடம்.
$200 என்னங்க? தேங்க்யூ.
தேங்க்யூ.
હે ચાલુ થઈ ગયું હો તારું. એટલે પકડ હું પેલા માં તારું ક્લિક કરું છું. નોટબુક લઈને બેસ. હું આમ નામ
સંજય લાલ સર નું નામ આવ્યું તો એમાં કર્યું.
બે તારી પેલી કાલવાળી લિંક પર ક્લિક કર્યું છે.
અમારા ક્લાસમાં કોઈ એમાં જોઈન નથી થયું.
કાલનું સેશન છે 4 થી 6 નું. શ્રુતિ કાલનું સેશન છે 4 થી 6 નું.
બસ માં
20 પતી જાય સુરેશ સર તો નીકળી જજે.
બેગ નો અવાજ આવે. આમાં મારા ક્લાસમાં કોઈ જોઈન નથી.
Good evening, people. I'm Karita Hapsipa, the course TA for the NPTEL lecture series on plants and bioprocessing. Welcome to the third session, which is week 3 course. We will discuss here some questions, mostly objective types and fill in the blanks, and brush up your knowledge on the week 3 lectures. So, so let's wait for 2 more minutes for people to join. Otherwise, we'll start solving the questions. I see some new names here in this series. I think this week course. So, can you guys introduce yourself? Should I call upon the names? Okay. Shruti Shah, can you introduce about yourself? Mr. Ashok?
Yes, good evening, all of you. Myself, Ashok Kadam, has just enrolled this the course that is plant cell bioprocessing, and I am from the Maharashtra Ahmednagar
Okay.
district, Ahmednagar.
Dipanvita, can you introduce yourself? She left the chat. Okay. So let's wait for 1 more minute. I think we can start. People who are going to join will be joining in the further few minutes. So I hope everybody has gone through the week 3 lectures. So the questions which I'm today I have 8 questions with me. All of them will be very easy if you have gone through the lectures. If you have any doubts regarding the week 3 lectures, you can ask me here in this forum. So I will be happy to answer. So let's go back to the first question, which is xylem elements are observed in torpedo stage embryos. Does anybody know the answer for this question? So I think last class we were discussing about different stages of somatic embryo. Today also we will brush upon that topic a little bit. So the statement here is xylem elements are observed in torpedo stage embryos. Is the statement given is true or false? I hope everybody has gone through the lectures. Can anybody answer? So the answer here is true. So what do you mean by torpedo stage embryo? What are the other stages of somatic embryos which are present? Can anybody brief out? I think we had discussed in the last class. So I'm sure whoever joined my last class will know by this time. And also you have gone through the lectures. What are the different stages which is present, which is commonly seen in somatic embryos? How many stages are there in a somatic embryogenesis stage or somatic embryogenesis, the or we can call somatic embryos? How many stages are there generally observed? No idea?
I think it is 8, ma'am.
8, or can you name them?
No, ma'am. I forgot it.
Okay, it's okay. So there are 4 different stages generally observed. There is the first stage which is, yes, Deepa, you are right. So what is the first stage? The first stage would be globular. Yes, right. Deepa is right. So the next stage is your heart shaped, followed by which torpedo stage, which is an elongated heart kind of stage, followed by which there is a phase. What does this mean? Heart and torpedo, yes. And then this is cotyledonary. So after this, we can see development of the entire plantlet from the somatic embryos. So the question here is xylem elements are observed in torpedo stage embryos, which will be here. Until this stage, xylem will not be observed in any of this previous stages, globular and heart-shaped um, somatic embryos. So in the torpedo stage, we can observe xylem elements, which will be so generally why xylem is used. Like what is the purpose of xylem in the plant cells? Plant. So the general purpose of having xylem in a plant tissue or plant cell is, the reason for the xylem is what? To conduct water or to conduct food? To transfer water or to transfer food?
To conduct the waters and the
Yes, yes,
minerals.
right. So xylem is mainly for the conduction of water along the different organelles of the plant body or system. Same goes with the cells. So as the different stages being discussed in the previous question, you can now answer this question, which is to identify the stage of somatic embryo development. Which one is this stage? So I just explained in the previous slide. What does this stage look like? No idea? It looks like torpedo for Monisha. Okay. Does it look like torpedo? Everybody says it is torpedo. It is D, cotyledonal, because you can see there is a leaf-like structure following, like forming there. If you go and check with the picture given there, in your plant cell bioprocessing slides also, you can see torpedo will be an elongated version, but here you can see a significant difference. It looks like a small plant emerging. So this is a cotyledonal stage. Okay, so I know it is confusing between torpedo and cotyledon, but it is a cotyledon. So this being discussed, okay. So let's discuss about somatic embryogenesis. Last session I have asked whoever joined to find out the applications of using somatic embryos. Have anybody found out? And I think you have also studied in this week regarding the applications of somatic embryogenesis. So I think those who attended the previous session also can answer, and those who didn't attend also can answer this question. So what is somatic embryos? What are somatic embryos? And why we have to do somatic embryo production or something? What application does it have? So what is somatic embryogenesis applied for generally? Why we have to go for somatic embryogenesis? No idea or don't want to talk? So I will list out then. Firstly, what we can do with somatic embryos. You can use it for micropropagation. You can go for improved secondary metabolic production. What else? What else you can think about the somatic embryos? No idea. So it can be used as model organism or a model to study the how the organs are being formed, its differentiation and regulation also. So how does the somatic embryos aid in micropropagation? Any idea? You are now aware of the different stages of somatic embryo. What does this stage signify? What does the stage mean? The way they grow, or what does it mean? Okay, so I think you have to next time when you join the session, you have to uh, come with a thorough understanding on the topic so that it can be more of like an interactive session where we can discuss your doubts also and also try to remember the topics which you have covered so far. So, okay, so you guys have to look upon the applications of somatic embryogenesis or applications of using somatic embryos. And okay, let's go back to somatic embryo only. So, any simplest way of making me understand what is somatic embryo? What does somatic embryo mean to you? What does somatic embryo mean? Apart from they are being used as synthetic seeds, right? I hope you have gone through the lectures on synthetic seeds and encapsulation also, cryopreservation also. Okay, we will go to the next. We will see how many of you are going to answer. So the question 3 is meristems can be used as explants to culture virus-free plants. The options given are A because they are sterile regions, B because of high rate of secretion of secondary metabolites, C. Because vascular connections are not yet formed. D. Because of the slow rate of plant metabolism. What is the answer for this question? So go through this question thoroughly. We will discuss further on meristem cultures. So what would be the answer for this question? What are meristems first? What are meristems, or where do you find meristem in plant cells?
At the tip of the plant, we got the meristems, and where the cells are divides fastly and helps in the growth of the plant.
Yes, you're right. Ashok, you're right. How many types of
Yes,
meristem are found generally?
there are three types of the meristems are there:
Yes,
intercalary meristem, lateral meristem, and apical meristem.
absolutely. You're absolutely right. So now you know about meristems and meristem cells, and these are faster growing cells. So can you give an answer on why these meristem cells can be used as explants for in vitro culturing of plants to get a virus-free plant? What do you find that this? What would be the reason?
I think pus is not there because they are the sterile regions. We cannot say it is sterile regions.
Yes, we cannot say it is
And
sterile region. It is still exposed to the outer environment.
yes,
So
and here the meristems is xylems and phloems are there. That's why C options is there. That is because vascular connection are not yet formed. I think there is a formats of the vascular connection. Either it is xylems is there or the phloems is there.
okay, you think meristem cells have xylem and phloem?
Yes, I think there is present
Okay,
xylems and the
so
phloems.
what would be the answer then for this question?
I think it is B or D. One of them is correct.
The answer would be C. I will tell you the reason.
Yes,
Because, of course, the answer cannot be A, still because the meristems are exposed to the outer environment.
yes,
And this question is about how to culture virus-free plants. So means there should not be any viral contaminants present inside the cells itself. So when you have gone through the lecture, you would have seen where meristem cells can be cultured because they have not yet formed the vascular connections yet. Because they are very rapidly dividing, they would have
yes.
not formed the vascular connections that fast. So that is the reason meristem cells can be used for virus-free plant initiation or culturing. So it is, I can put it like, if you select the meristem tissue for the culturing an in vitro plant, it is most probably will be free from any virus, because when you took the meristem cells, it would not have formed any vascular connection, so it doesn't have any connection with the other parts of the plant vascularly. Okay,
Okay, ma'am.
the
Okay.
cells will be very fast, the division rate will be very fast, so that they don't uh, compare to the formation rate of your vascular connections. That's why there will be
Okay.
less um, chance of viral contaminants present. Okay, and viruses also cannot transfer very quickly through your plasma desmata, which is present in your cells. That is also the reason. Thank you for answering the question.
Okay, ma'am. Will.
So the next question is about the cryopreservation. Why do we do cryopreservation?
It is a process like micropropagation, but in the micropropagations, we culture the plants from the explant
Yes.
okay, healthy virus-free plant. But in cryoprotectant, I think it is related to cryopreservation methods is there,
Yes, yes.
and that small cells are the preserved. And I think here is methanol is using non-permeating as materials in that.
Uh, Whether you think methanol is a non-permeating cryoprotectant, non-permeating in the sense it won't penetrate the cells, or it is a permeating one. Permeation means penetration inside the cells. That is what it means in the question.
I think it's non-permeating.
It's a non-permeating. But have you gone across this methanol causing? Uh, so you are from science background, sir?
Yes, ma'am.
Uh,
Science background.
yes, you are a BTech or MTech or BSc
I am MSc Botany, ma'am.
bot.
MSc Medicinal Botany.
Okay, okay. So you would have done extraction and all, plant
Yes,
extraction, tissue
yes,
extraction.
yes.
So this methanol you generally use for extractions, right?
Yes,
Yeah,
extractions we use the different
yeah,
solvents like
different
methanol,
solvents. Yeah, sure.
petroleum, ethers, all solvents are there to extract the plant.
Yes. So, do you think methanol is a non-permeating one? Forget about the cryoprotectant, because methanol is a solvent which will cause a certain degree of damage to your cells, right?
Yes.
So that is why you are able to get your extract out or anything present inside out. So if you apply that here, do you think the answer would be true or false? Because here in the statement it is written methanol is a non-permeating cryoprotectant.
Methanol is a permeating cryoprotectant, and this is a false, I think.
Yes, you are absolutely right. So the problem is methanol will harm, generally will harm the um, any alcohol will cause certain level of damage to your cells, which is specifically the plant cells. So the question asked here is with even though it is with respect to cryoprotected, methanol is generally a permeating one. So even in the form of solvent. So methanol, the answer for this is false, which is the statement would be methanol is a permeating cryoprotectant. So, right, sir, your answer was correct. The answer is false for this question. So, what is the role of mannitol or sucrose in cryopreservation? So, I hope you have all gone through this video lectures on cryopreservation. I think it is 8th or 9th, yeah, 10th lecture, I think. So, can you brush up on those topics and try to remember what is the role of mannitol or sucrose in the cryopreservation? Mannitol or sucrose, they both are sugars, right? So, what would be their role in cryopreservation? Take your time, think about it, and answer. Julie has answered the improve cell growth. Okay, we will see. What would be the other answer? Um, they improve cell growth. No. So, what does cryopreservation mean? Cryopreservation is preserving something in under at what temperature? Let's go to that. What temperature? At what temperature cryopreservation is being carried out, and what does cryopreservation mean? What exactly is being done there? You guys take 2 minutes and try to answer this question. What is cryopreservation? At what temperature it is being carried out? What do you do in cryopreservation, Siddharth? To preserve the sample at low temperature, yes. At low temperature means how low temperature it is. 4 degree, 0 degree, what degree it is. How low you can go.
I think it is a minus 180 or 170
Minus
nearly.
180 or 170. No, it
It
is
is low
way
temperatures.
more lower than that, sir. Almost freezing. Yes, what Jui is telling that almost freezing temperature means. What do you mean by almost freezing temperature, Jui? Is minus 20 freezing enough or minus 80 we have to go or beyond that? So cryopreservation is usually done it around. So when you remember that, how do they do cryopreservation? What is the main reagent which they use in cryopreservation? If you remember that, I think you will be able to answer the question at what degree. What do they use to take them to the freezing temperature? Nobody remembers. Heard about liquid nitrogen? Liquid nitrogen? So they use liquid nitrogen to take the cells to or store the cells in lower temperature, which is the temperature of which would be what? Sorry. What is the temperature of liquid nitrogen? Any idea? Forgot completely. Cryopreservation is done using liquid nitrogen. So at what temperature? Minus? Minus how many degrees? Minus 196 degrees Celsius. So this is the degree which the liquid nitrogen will be stored. Like liquid nitrogen, have you guys come across that? Even nowadays they are selling in the ice creams with liquid nitrogen at all. Have you come across those? It will come like smoke. A bit of liquid nitrogen, if it falls on something, it turns hard and you can easily break it. So this liquid nitrogen cryopreservation is generally done using liquid nitrogen along with some reagents which will prevent um, the cells from death actually. So cryopreservation is being followed. The major purpose is what? The storage is at minus 196 degrees Celsius because the metabolic activity will be ceased. So all the metabolic activity which is going on in the cells, like production of some compounds or a division, everything will stop when it is being stored as minus 190 degrees, 196 degrees Celsius, around 180 to 196 degrees Celsius. So when you take these things, cells, out of your preservation mode, you can thaw them back, which is bringing back to the room temperature, and putting these cells in a newer food source or new media, it will then start growing. It will start regaining its metabolic activity again, and then it will behave like normal cells. Of course, there will be some loss due to freeze storage, um, but in this way, you can store more. What we can call, we can store the explants which are not easily available, or we can use this for germplasm conservation, where you want to conserve some species which is no more and which are on the verge of extinction. Those things or those plants, their cells, their seeds, etc., can be stored via cryopreservation. So that is one major advantage of using cryopreservation. So coming back to the question. So here the question is about mannitol or sucrose. So the answer is they act as osmoticum. So what does osmoticum mean? What does osmoticum mean? So mannitol or sucrose, they do not decrease, they are not used because they are decreasing cell toxicity, and they are also not used because they improve cell growth. When you are freezing it, why there is a purpose of adding cell growth initiating things like mannitol or sucrose in the media? It is not necessary, right? Because you are freezing it, the very same purpose is to cease or stop the metabolic activity of the cells, so that it is frozen as such. So there is no purpose of using them for improving the cell growth, and there is no purpose of them to be used for reducing the contamination because they are sugars. And what else? So the answer is, I have told the answer. The answer is they act as osmoticum. Now tell me, what is osmoticum and what is the purpose of using an osmoticum while storing the plant cells? So basically, what and all will be there in your, say for example, you have a vial, a tube. In this tube, you have your glycerol. Glycerol is generally added along with the DMSO or some other solvents which are cryoprotectants in nature. Along with your cells, they are putting mannitol or sucrose also. Then they are storing it in a big tank, liquid nitrogen. This is how cryopreservation is done. So why this mannitol or sucrose is used? I told the answer. It's osmoticum. So my question is, what is osmoticum and why we have to use it in freeze storage or cryopreservation? Any idea? Osmoticum, what is the meaning of osmoticum? Have you come across osmotic pressure, osmosis? I'll give you, yeah.
Yes, it is a substance or the which increase the osmotic pressure in the cells
Okay, it is
or
a
the
it is a substance which acts to help in maintaining osmotic pressure in the cell. Okay, so why you have to do that? Why you have to give an osmoticum or osmotic solution to the media or to the cryopreservation vial? Why do you have to add it? Any idea? Have you gone through what is that? Protoplast culture and all? Why you add your osmoticum there? The same reason here. In protoplast culture, why you add osmoticum? To prevent your cells from breakage, right? What will happen if you put, you're right, the answer is it is a substance which acts to supplement the osmotic pressure in the cell. So if you put the cell in water, what will happen? Cell lysis will happen. If you put that in a sugar or mannitol or sucrose, what will happen? The concentration will be what? It will be,
I think if you put in the mannitol, the osmotic pressure will be increased.
pressure will be increased. Then what will happen?
Movement of the solvent is there, or movement of that liquid molecules from higher
Okay,
concentration
to
to
lower
lower.
concentration. Okay. So this mannitol or sucrose, mannitol and sucrose, they both are, either of them will be used in cryopreservation so that it will prevent your cells from undergoing shock and cell death, because you are freezing it at very low temperatures. uh, the water will start freezing there inside, right? Any liquid in the your vial will start freezing at the time when water, everything, the solvent, everything is freezing. This concentration gradient will be aided by this osmoticum, which is the solution which you are adding to maintain osmotic pressure inside the cells, will prevent or the damage caused due to this variation in the osmotic pressure. That's why. So the sixth question is about secondary metabolites. So which of the following is a non-protein amino acid released by plant as a defense mechanism, which replaces amino acids in the metabolism of herbivores? So the option A is glycine, option B is cannabinine, option C is arginine, and option D is proline. Yes, the answer is cannabinine. So what else do you guys know about cannabinine? What does this cannabinine does actually? Why the plant produces this cannabinine actually? Why it is being produced? It is a plant metabolite, right? It is being produced by the plants as a defense mechanism, understood. And why does this plant produce this particular metabolite for defense mechanism? Sorry.
I think it is defensive compounds, ma'am, which is the producer in the plant bodies, mostly in the seeds.
Yeah, it is mostly produced by leguminous plants and their seeds. Sorry. So, what does it do? It is as a defense mechanism. What does it do to the herbivore? It resembles. It will be resembling to any particular amino acid which is being produced by the animals. So this cannabinoid is being produced by certain plants, especially seeds of some leguminous plants. And when an animal or a herbivore is what something which feeds on a plant or plant-based food, which is some animal or something is which is eating plant-based food. So when this, say for example, deer, if it comes and consumes this seed or plant material which contains cannabinoid, what will happen? This will,
It is,
yes,
it is a toxic, ma'am. It is
yeah, it is
toxic to that
toxic.
animals.
Yeah, it is a toxic metabolite. So that what will happen? So what happens is this particular metabolite resembles very similar to your amino acid arginine. So this will go and replace the arginine in the herbivorous animals system. It will cause a lot of what you call, like you said, it will be toxic to the system, so that later on it will not be affected much. So instead of arginine in the animal's body, it will be cannabinoid which goes and sits in that place. So that will cause, because of this amino acid replacement, there will be structurally different protein forms which is formed, which will further aid in the toxicity of the toxicity to the particular animal. So that is the mechanism of action. How this cannabinoid is being replaced into arginine in the animal system. So it is produced as a defense against that particular plant. So if next time the plant won't be consumed by that particular animal, right?
Yes.
Yes. So the answer would be cannabinoid. So sorry. So we were talking more about secondary metabolites. So what are secondary metabolites? Can anybody answer? What are secondary metabolites?
Secondary metabolites are produced in the plant bodies
Okay.
from the primary metabolites like phenolic compounds,
Okay.
amino particular amino acids, and main functions of that secondary metabolite to depends from the herbivores plant bodies.
Okay. So according to you, what is primary metabolites then?
For the metabolic activities of the plant, it is produced inside the plant bodies.
They both are produced inside the plant bodies only, na sir? Both primary and secondary metabolites. And how do they both differ? So the actually the primary metabolites are which is the metabolites which are necessary for the growth of the plant, which include any protein, amino acids. These are all needed for the plant's growth. Nucleic acids like your ribose, ribulose, what else? Um, adenine, guanine, thymine, everything. So all the nucleic
Yes.
acids, everything. These things and all necessary for the growth of the plant. So these are primary metabolites. These will be produced during the growing phase of the cells. And secondary metabolites, these are not generally much, they do not have any obvious role in the normal metabolism of the plant. They are not produced during the growth phase also. They will be produced during the stationary phase, and they will be mostly, like you said, sir, it will be for, they have roles other than yeah, they have roles other than being um, needed for the
Growth.
growth. So
Yes.
the example, like you said, it will be for defense mechanism or to attract pollinating insects or something, pollinating animals and
Yes. Yes.
seed dispersing
Ecological
animals.
functions.
Yes,
This secondary metabolites show the ecological functions in the
yes,
bodies.
yes. Ecological functions also, and they have also access interacting elements and what else you can call for defense also. So these are some of the can you so okay. So we are talking about secondary metabolites and how how do we instead of primary metabolites, how do we find the secondary metabolite useful for us? So these generally doesn't occur in higher qualities, like they will be present in relatively low quantities compared to your primary metabolites because they are needed for growth and these are produced only in the case of any attack or some defense kind of any need at that time only these are being produced. So how if you want to produce some secondary metabolite, what you will do? You have to culture the cells and produce something like that. So what you will do when you will select the cultures when they are in which phase? See, for example, you need you are cultivating plant cells, so you need secondary metabolite. So you are allowing the cells to grow for some time period, say example up to 20 days. And let's see, this is biomass or weight we can keep for easy understanding. So from the starting day, slowly, your weight of the biomass or the weight of the cells in the media will start growing. Assume. Let's assume that we have done this experiment. So what happens after a certain point? It will reach a stationary phase where there will be no cell growth observed and there will be a decline phase, deceleration phase. Okay. So this is you would have learned this in microbial growth kinetics. So this phase is lag and this is log, where log is an exponential growth phase. So as I have told here, this is this phase, growing phase, when the cells will be in growing stage, rapidly dividing. So in this phase, we can look for primary metabolites. But stationary phase is a phase where the cells are compromised, they are under stress. The number of growing cell is equal to the number of death cells going for death. So this is the stationary phase. This is the phase where you can study or you can observe most your secondary metabolites. So if in case you want to produce secondary metabolites using in vitro cultures or in vitro cell system, you have to uh, select the cells when they are in their stationary phase. Did you get the point? Okay. So before this, I wanted to discuss something. So, so I wanted to discuss about when this cryopreservation thing. So what are synthetic seeds actually? So what are synthetic seeds actually? They are somatic embryos, of course. They are what are the different four types? You would have come across four types of how they call coatings or what the type of encapsulation. So what are those? Really? How many types and what are the types? So what are the different types of somatic embryo encapsulation? Do you remember coated desiccated somatic embryos, uncoated desiccated somatic embryos? Do you remember? These are the four different types of um, somatic embryo encapsulations. So this is how four different ways you can, four different types where you can synthesize your synthetic seeds. So what do these things mean? What is uncoated? What is coated? What is desiccated? What is hydrated? What does it mean? Nobody knows the answer. Okay, next session you have to give me the answer for this question. So last week I have asked about the applications of somatic embryogenesis. Did anybody find the answer? Nobody did. It's okay. Just go through the lectures once in a while so that it will be um, helpful for you to understand the subject better. So whatever doubts arises when I'm discussing something here, you can ask here. Okay. So we will go to the next question. Which pathway leads to the production of the plant hormone jasmonic acid? What is the answer for this? What is the answer for this question? So, what pathway leads to the production of the hormone jasmonic acid? What is the answer? Which pathway it is? No idea? Heard about octodecanoic acid or octodecanoid pathway? Sorry. So, what does this pathway do? Yes, that it produces jasmonic acid. What is this jasmonic acid? What's its role? It is being produced by plant, so it is a plant hormone which is jasmonic acid. So, what is the purpose of this jasmonic acid? Why does the plant produces this jasmonic acid? Any idea? You are right. It is octodecanoid pathway. Thank you for answering.
I think it is the growth regulating substance or the
Growth
hormone
regulating
in a
substance.
higher plants.
Okay, you think it is a growth regulating substance. Okay. What else? Any other answer? It is a secondary metabolite or it is a primary one?
Secondary. It
Secondary
is
metabolite.
secondary.
Okay. So this jasmonic acid, as you pointed out rightly, it is it's used as a regulatory hormone for growth, and also it is a compound which is generally involved in the cellular defense and development in plants. So it acts. I think in the further um, sessions you will be uh, getting lectures about induced resistance or systemic acquired resistance of plants. How do they acquire resistance from any particular insect or uh, damage or something? There you will be getting more insights on this jasmonic acid. You're right, it is a growth regulatory hormone and it is also used for defense purposes from by the plants. So the answer to the question will be what? The release of secondary compounds by one plant to have any inhibitory effect on the neighboring plant is referred to as dash.
I think it is allelopathy. In
Al-
allelopathy, one plant effect on the other plant's growth.
okay, in allelopathy, yes, you are right, sir. The answer is allelopathy.
Okay,
So
ma'am.
here, the release of a particular compound or any in the form of gas or any by compound or something by one particular plant will have any inhibitory effect on the neighboring plant. which is like it will not let the other plant grow, or it will cause some damage to the other plant which is nearby to it. So this phenomenon will be called as cellulopathy. You are absolutely right. So, so as we have discussed all 8 questions today, I would actually I have asked some questions related to the enzymes which will be used for breaking cell aggregation or breaking the cell wall. Had anybody gone through it and found answer? One, I gave the answer which is pectinase. And do you think there are some other enzymes which are present with which you can break the cell wall of a plant cell? Cellulase, okay, pectinase, cellulase, what else?
Oxidoreductases or
Why,
lyases may be there, which
how
breaks
does
the cell walls.
lyases, in the sense these are categories of enzymes you are talking about? Can you name any specific enzymes, sir? So if you think about the components present in the cell wall, say for example pectin is present, so if any enzyme which can degrade the pectin, which is pectinase, if it is added, it will break the cell wall. So cellulose is also present, so if you add cellulase, it can break the cell wall like that. There you have some examples. I think it will come with the protoplast technique where you will get this enzymes to remove the cell wall, if I'm right. So in your protoplast technique, there will be some enzymes mentioned actually. Pectinase, hemicellulase, mesozymes, diaslase. These are some of the enzymes which are used for um, breaking the cell wall and disrupting the cell wall so that it will aid in um, single cell single cell cultures and also in protoplast techniques. So I was talking about protoplast. So what does a protoplast mean and why it is used? What does protoplast mean?
In simply, the protoplast is a cell whose the cell wall is removed.
Yes, sir. The cell wall is removed, and only a cell without a cell wall is the protoplast, right?
Yes, yes,
So,
yes.
what is the purpose it is being used for? Any idea? Without cell wall, yes, Deepa, it is correct. Without cell wall, a cell without a cell wall. So, any reason why we are doing? Because it is a very fine technique where you have to use microscope and have to be very careful not to break the cell. So, why we are going with protoplast cultures? What is the necessity? Any answer which comes in your mind? It need not be the right answer. You can, whatever you think, you can speak here. That's why it is an interactive session.
I think for study the cell or formations in the plant.
That also a way of putting it together. It can be used as a model system to study the cell wall, how it is formed, those things, and role of each element. And have you thought about genetic transformations? with the cell wall being gone, you can do genetic transformations very precisely, transferring your external DNA or directly inside. You can do somatic cell fusion. From a single cell, you can even get a clone to get from a single cell, you can even generate a entire plant using single cell cloning techniques. Somatic hybridization you can do. What else? That's all, or is there something more?
I think for the DNA transformations, we use
Yes,
that
it
protocol.
is the genetic transformation which we talked about for to modify the genetic makeup of the living cell. We can do protoplast fusion or somatic hybridization. Without the genetic transformation, somatic hybridization can be done just by mixing protoplasts of two different type of cells or two different species of plant cells. You can mix it. You can cause hybrid between those two by using somatic hybridization and then develop them into an entire plant also. That is also possible. So these are some of the advantages of using protoplast culture. So now coming to, have you come across germplasm conservation? Your entire topics on week 3 is about the synthetic seeds, somatic embryogenesis, so and production of secondary metabolites and all. So we should also think about germplasm conservation. What does this germplasm conservation mean? Means what is germplasm conservation? It has to do something with all the topics which we have covered today.
Used for the conserve the genetic traits of a particular species.
Yes, you are right. So to conserve any particular species which might go into extinction or we, God forbid, we don't know what will happen in the future. Some species might be already extinct or on the verge of extinction. So, or even the common species which we think won't be going into extinction soon. Extinction soon. So, there are two different types of preservation. Can you name it? Germplasm. In germplasm conservation, there are two types. One is in situ preservation, and the other one is ex situ preservation. So, what is in situ preservation and what is ex situ preservation? In situ, yes. In situ, yes. Jose is right. In situ and ex situ. So, what is meant by in situ preservation? Any example? In situ is in lab. Then what is ex situ?
Uh, I think national park is examples of in situ conservations
Okay.
and cryopreservations. Just we have taught it is comes under the ex situ conservations.
Yeah, you are right. Absolutely right. So, in situ is in their natural habitat itself. You are preserving them in their natural habitat itself. Example like sanctuaries or preserved areas or what you call um, where nobody is allowed inside is like by the government. Nobody is allowed inside, which might, because people might harm the plant species which are being cultivated there or which are present there, and those areas will come under restricted categories, and the ecosystem will be preserved. So, national parks, sanctuaries, wildlife conservation centers, those things and all comes with in situ preservation. Ex situ preservation is, like you said, sir, it is um, in the form of seeds or in the form of in vitro culture systems. You can continuously keep them cultivating or culturing them in the lab in the form of plantlets or a cell suspension or cell systems or even cryopreservation. So all these things will come under ex situ preservation, where the genetic makeup and of the entire cells will be preserved there. Um, so these are the contents which I wanted to share with you guys today. So did you guys receive at least this time the drive link? Anybody, can anybody answer? Did you guys get any drive link with the PPTs? Not yet. You didn't receive any link from NPTEL which had PPTs or something? No. Okay, then I'll
No
just.
link,
Yeah, it's been
no
already
link,
weeks.
no
Okay,
any link from the NPTEL.
no link. No link stating this is the drive, not like that. Okay, okay, I'll check once. So, do you have any? I hope you have submitted your first assignment. It's due it or it's done already. Done already.
Yes,
Okay,
ma'am. Yes,
okay,
ma'am. Done
okay.
it.
Sure, sir. Fine. So, if you have any doubts, you can ask me here. Not about the assignments, but about the week 3 lectures. Um, if you don't have any other doubts, you can now leave the call. You can ask me any doubts. If you have any other doubts, not if the doubts are not arising now, you can always put it in the discussion forum, mentioning the date of the lecture and mentioning it as from the live session. Thank you for joining.
No, ma'am. No any doubt.
You don't have any doubts? It's okay, sir. If nobody has any more doubts, you can leave the session. We can close the session now. Dear students, thank you for joining the session. You can leave the call. Um, we have already closed the session. If you have any doubts, please post it in the discussion forum mentioning today's date. So we will meet you in the next session, which will be held on the next week, Friday, 6 to 8. Happy learning. Thank you.